A potential site of action, the thalamus. The regional interaction between the thalamus and anesthetics supports the proposed localized thalamic consciousness switch. Such a switch mechanism may be a central component of a broader dose-related anesthetic cascade of events. At the cellular level, anesthetic agents compromise the natural firing patterns of thalamic network neurons, such as thalamocortical, corticothalamic, and reticulothalamic cells by hyperpolarizing their resting membrane potentials. As a result, and in a manner that parallels the mechanisms underlying psychological sleep, a greater portion of these network cells experience bursting rather than tonic activity. This, in effect, blocks or diminishes synaptic transmission of sensory information through the thalamus and diminishes the high-frequency rhythms that characterize the spontaneous activity associated with the awake state and dreaming mentation. An example of the dose-dependent ability of isoflurane to reduce the reliability of sensory transmission through the sensory thalamus is shown in the Detch et al. Experiment, who recorded thalamic unit activity in the rat following a temporary 100 Hz stimulation of mechanoreceptors. The thalamic units tonically followed the stimulation at low isoflurane levels, but would show only an initial burst at the stimulation when the isoflurane levels were higher. This finding is consistent with the thalamic consciousness switch hypothesis, as it shows that thalamic unit activity switches its firing pattern in a reversible manner with increasing anesthesia dose. Further work by this group suggests that the decrease in thalamic firing rates that occur at higher anesthetic doses is likely due to a decrease in excitatory corticothalamic feedback mediated by both glutamatergic and GABAergic effects. This suggests that the switch in thalamic unit activity is driven primarily through a reduction in afferent corticothalamic feedback and is not necessarily a direct effect of anesthesia on the thalamic neurons themselves. The cellular mechanism or mechanisms through which a thalamic consciousness switch might work remain unknown. Two facts converge to suggest that neuronal nicotinic acetylcholine receptors are a plausible anesthetic target. First, neuronal nicotinic acetylcholine receptors are potently inhibited by many anesthetics at a sub-anesthetic dose. This fact suggests that nicotinic acetylcholine receptors are inhibited in significant proportions at the concentration of anesthesia associated with a loss of consciousness. Second, the alpha-4 beta-2 subtype of the nicotinic acetylcholine receptor has its highest density of expression in the thalamus, suggesting that the localized decrease in regional thalamic activity seen in anesthesia brain imaging studies might be due to a regionally localized antagonism of nicotinic acetylcholine receptors and might really be a reflection of a localized direct action of anesthetics on the thalamus and not just a secondary reduction caused by decreased corticothalamic activity. Alkire et al. Directly investigated whether thalamic nicotinic mechanisms might play a role in mediating the unconsciousness component of inhalation anesthesia. This idea was tested in sevoflurane anesthetized rats that were given enough sevoflurane to induce a loss of the writing reflex, otherwise known as LORR, a correlate of unconsciousness. With rats rendered unconscious, minuscule amounts of nicotine were microinfused into the thalamus of each rat through a previously implanted cannula that was aimed at the central medial nucleus of the thalamus. The CM is an important component of the intralaminar thalamic nuclei, and it represents the rostral extension of the ascending reticular activating system, otherwise known as ARAS. Previous work by Miller and coworkers had discovered that the CM thalamus plays a role in arousal and seizure propagation. Miller found that microinfusions of GABA agonists into the CM thalamus would cause a rat to lose consciousness, often within 90 seconds. Nicotine microinfusions localized to the CM thalamus resulted in rats awaking from anesthesia, despite still being in the middle of a chamber filled with anesthesia. The case for the thalamic consciousness switch would seem to be strongly supported by the nicotine reversal of unconsciousness experiment. Importantly, however, Alkire and colleagues also tried to induce unconsciousness with intrathalamic microinfusions of mecamylamine, a nicotinic antagonist. If thalamic nicotinic mechanisms were causal to anesthetic-induced unconsciousness, and the thalamus was a switch site mediating the consciousness-off component of anesthesia, then intrathalamic mecamylamine should rapidly induce a loss of consciousness. This would be a similar experiment to that reported by Miller and colleagues with their GABAergic agonists. Mecamylamine, however, did not induce a loss of consciousness. The overall pattern of results where the agonist reversed the unconsciousness producing effect of anesthesia and the antagonist failed to cause a loss of consciousness suggests that the intralaminar thalamic nuclei may act more as a consciousness on switch than a consciousness off switch.
Another possible site of action is the cortex. Even with rapid fMRI imaging, brain scans are too slow to determine whether the thalamus or the cortex shuts down first. This could be important because if it is the thalamus that is regulating awareness, it could be targeted to produce selective changes in consciousness. Here are two studies that attempted to localize consciousness. The first one, by Lukács and McIver, tested anesthesia on GABA receptors in cortical slices in vitro. Their findings support an anesthetic slowing of cortical activity. Since this study was done independently of subcortical structures, it shows that the cortex itself can respond to anesthetics, and it isn't just modulated by the thalamus. Henske and colleagues measured the in vivo change in cortical firing rates in rats after exposure to increasing doses of anesthetics. They then correlated these changes in firing rate with the concentrations of anesthetics that were used to increase GABAergic currents in vitro. They found a correlation between the suppression of cortical firing rates and anesthetic effects of GABA currents, which indicates that the cortex is responsive to the effects of anesthesia. Finally, Veli and colleagues recorded cortical and subcortical EEGs in Parkinson's patients undergoing anesthesia. They found the anesthetic in the cortex determined consciousness, while anesthetic in the thalamus determined whether patients would move with somatosensory stimulation. These findings, combined with those from the other studies, support the idea that the cortex is turned off before the thalamus. Many hypothalamic systems are affected by anesthetics. The limbic system may regulate the arousal-suppressing effects of anesthesia, and midbrain areas can induce unconsciousness when injected with barbiturates. However, the total effect of anesthetics likely involves a complex network of interactions.